Hi guys, in this video I'm going to be doing an experiment looking at osmosis occurring in potato cells. What I've done is I've made up a whole heap of primary solutions here with different amounts of sugar in them, going from 0% sugar to 50% sugar. So this is 50 grams of sugar in 100 grams of water. And I've gone 10, 20, 30, 40 between that. I've then set up in each of these little beakers potato that I've cut up small. So I've got around 5 grams of potato, but I've cut it up into very small pieces to increase the surface area. Now what I'm going to do is for each one of these beakers, I'm going to pour the corresponding solution into them, and I'm going to put in about 80 mils into each beaker. I'm then going to leave it for 24 hours and come back and see what's happened. What I'm hoping to see is that the potato in the 0% solution will increase in weight because the water will go from the solution into the cells of the potato through osmosis, and my 50% solution will actually lose weight because the water will go from the potato cells out into the solution, because it will be a lower concentration. Remembering that osmosis is the movement of a solvent from high to low concentration. So I'll just fill these up. There we go. Okay, now the first thing that I'm seeing here is that the 0, 10 and 20% solutions, the potato is sunk at the bottom. Well, actually in the 20, a couple of them are floating and a couple of them are sunk at the bottom. But the 30, 40 and 50% solutions, the potato is floating on top. And this is because of the higher amount of sugar that is dissolved in the water, therefore increasing the density of that water. So what I'm going to do now is leave this, come back tomorrow and see how we go. Alright, so I'll come back the next day to check on my potato cells. These have been soaking in different concentrations of sugar solution overnight. Remember, we've got our 0% at this end and our 50% at this end, which is behind the scale, so you can't actually see it. And what I've done this time is I've actually put Petri dish covers over top of each beaker. This is to stop bacteria and fungi growing on the top, because I've been fallen into that trap before, so I can just take those off now. Now what we want to do is get rid of the water and weigh the potato cells. So I'm just going to start with that 0% solution. So this is all distilled water. Pour as much of that off as I can. Just all of it, excellent. I'm going to get a piece of paper towel and just getting all those pieces out. I'm going to get a stirring rod. Okay, so stirring rod will make this a bit easier. Get those out. Wonderful. So the paper towel is going to soak up all the excess water. Then I'm going to, so just drying those, I'm then going to place these pieces of potato into the Petri dish on the scales. Now this was a 0% solution, so the potato is hypertonic to the water surrounding it. So what we're hoping to see is the potato increase. So we started, and this one was 5.04 grams that we started with. So we're hoping to see it go a bit more than that, and we got up to 5.87 grams. Okay, so that's pretty good. I'll just make a note of that here. 5.87. Okay, and I'll put those back into the beaker. I don't really need those for anything else, but save the packing up later. So get my Petri dish wiped. Now what I'm going to do, I'm not going to go through all of these and bore you. I'll just go through the 50% one. So this solution was 50 grams of sugar in 100 or grams or 100 mils of water, whichever way you want to look at it. This is a solution where the solution is hypertonic and the potato is hypotonic. So what we should see is osmosis moving the water, the solvent, in the direction from inside the potato to outside the potato. So I'll put this on my paper towel, give it a bit of a pat dry, and put it in my Petri dish. Okay, so this one, hoping to see a loss in mass. This one started out at 5.11 grams. So hopefully it'll go down. Fingers crossed. Beautiful. 4.18 grams, that one. Okay, what I'm going to do now is just go through the rest of them and measure them out. Alright guys, so I've crunched some numbers and here are my results. You can see those ones that I actually took on the video. The 0% concentration, or the straight distilled water, went up to 5.87, started at 5.04, and the 50% started at 5.11 and went to 4.18. So what I've done in this column is I've worked out that change in grams, and then I've converted the change in grams into a change as a percentage. And I did this by taking this change in grams and dividing it by the initial mass. And the reason I did this is because you've got a different start masses here. We need to somehow sort of smooth out those numbers. So I put it into a percentage to do that. And you can see that the 0% solution, we had a 16% increase, remembering that the potato is hypertonic to the solution around it. Therefore, the water is going to go into the potato to try and even out that. And the 50% solution, we've got the potato being hypotonic to the water around it. So the water leaves the potato through osmosis, and we get a decrease in that mass. Now you can see, actually I'll put it into a graph to make it a bit clearer. You can see that we had a very clear trend for these first couple. So that was about 17% increase. We've got a 4% decrease and a 20% decrease for those first three solutions. The solutions after that, 30, 40, 50, tend to then flatten out. Now I'm thinking the reason for this is probably because I only left it for 24 hours. There's basically a balancing act here with leaving it for long enough that it reaches equilibrium and not too long that the potato starts rotting and falling to bits. So maybe if I'd done it for 2 days, I would have got some better results here. But we can definitely see this trend here. And if I were to have those better results, we'd actually be able to work out the concentration of the sugar or starch, okay, let's just say carbohydrates, found inside the potato. And for my results, it's about 15%, but as I said, that's skewed by these ones here. And that is the end of this prac. So we've seen that osmosis occur in both directions depending on the concentration of the solution and the concentration inside the cells. Now I talked about the equilibrium point that's been reached here before. When we're talking about solutions, this would be called isotonic, okay, where both solutions are the same concentration. That's pretty much the whole prac, guys. Thanks for watching. Peace out.